The article, titled Schwannosis in the Brain of a Neonatal Calf, authored by Diana Bokinska and Sharp, Brian Toland, and Alina Demeter, reports a rare case of schwannosis in a one-day-old Holstein-Friesian calf. Schwannosis is a non-neoplastic condition characterized by the proliferation of Schwann cells within the central nervous system, accompanied by attempted myelination. This particular case is noteworthy because schwannosis is rarely documented in veterinary medicine. The calf, part of a larger herd, was submitted for postmortem examination due to the presence of arthrogryposis, which refers to contractures of the joints and premature death. Upon initial examination, no other significant gross abnormalities were observed. To identify potential infectious agents, tissue samples from the liver, lung, and brain were collected and tested. The testing included screening for bovine herpes viruses 1 and 4, bovine viral diarrhea virus, Schmallenberg virus, Anaplasma phagocytophilum, Campylobacter fetus, Chlamydia abortus, Coxiella bernicii, Leptospira species, and Listeria monocytogenes. Additionally, the abomasal content was subjected to both anaerobic and aerobic cultures. All the tests for infectious agents yielded negative results, and the cultures remained sterile. A comprehensive histopathological examination was performed on various tissues, including sections from the brain, cervical spinal cord, liver, lungs, kidneys, and heart. Microscopic analysis revealed multifocal proliferations of spindle-shaped cells, predominantly located around blood vessels within the white matter of the medulla oblongata and, to a lesser extent, in the gray matter of the midbrain. These cells, measuring between 7 and 15 micrometers, exhibited eosinophilic cytoplasm and oval nuclei. Furthermore, they stained faintly blue with Luxol fast blue. No other notable histological changes were detected in the central nervous system or other examined organs. Immunohistochemistry was employed using formalin-fixed, paraffin-embedded tissue sections from the affected brain areas. The antibodies used included periaxin, myelin protein 0, SOX10, S100, and vimentin. The proliferating spindle cells displayed diffuse cytoplasmic immunolabeling for periaxin, diffuse granular membranous and cytoplasmic immunolabeling for myelin protein 0, granular intranuclear immunolabeling for SOX10, intracytoplasmic immunolabeling with S100, equivocal immunolabeling for vimentin. Based on these histological and immunohistochemical findings, a diagnosis of schwannosis was established. Schwannosis involves the invasion and proliferation of ectopic or aberrant Schwann cells from the peripheral nervous system into the central nervous system, accompanied by attempted myelination. This condition is rarely reported in veterinary literature. A search of several databases revealed only one prior publication describing schwannosis in a calf and 3 foals. In that report, the animals exhibited spinal schwannosis along with hydrocephalus or scoliosis, syringomyelia, syringohydromyelia, and small or asymmetric gyri and folia. In the current case, the calf presented with arthrogryposis and tested negative for common perinatal infections. The underlying cause of schwannosis in this particular case remains unknown. However, the authors propose that it may have contributed to fetal paralysis and the development of arthrogryposis. They also discuss the potential origins of Schwann cells within the central nervous system, including migration of ectopic peripheral Schwann cells during development or differentiation of resident brain oligodendrocyte progenitor cells. Another theory suggests that schwannosis may be a reactive response to nerve injury or chronic inflammation. This case emphasizes the importance of considering schwannosis as a differential diagnosis in calves presenting with arthrogryposis when infectious causes have been ruled out. Additionally, the article briefly touches on other Schwann cell disorders, such as schwannomatosis and schwannomas. Thank you for listening to Vet Neurojar. Keep those minds inspired, hearts light, and tails wagging.